Welcome to G Fun Facts Online. Today we are stripping away the macro-level view of human aging and zooming deep into the microscopic molecular triggers that dictate the exact pace at which our cells decay. For decades, the quest for a longer human health span has focused on sweeping systemic changes like caloric restriction, cellular reprogramming, and the clearance of senescent cells. But at the very center of this biochemical clockwork lies a single, elegantly simple molecule, the amino acid tyrosine. You might know tyrosine as a benign dietary supplement, highly prized as the foundational building block for the catecholamine neurotransmitters, dopamine, norepinephrine, and epinephrine. It is famous as the molecule of alertness, drive, and metabolic baseline. However, modern gerontology and molecular biology have uncovered a staggering and far more complex reality. Tyrosine is not merely a structural component or a simple neurochemical precursor. It is the master regulator of cellular communication. Through a fundamental process called tyrosine phosphorylation, where phosphate groups are strategically added or removed, this amino acid acts as the ultimate biological on and off switch. It governs the massive signaling pathways that command a cell to divide, to enter senescence, or to trigger the aging process itself. This brings us to a profound longevity paradox. While tyrosine is absolutely essential for the neurological vitality we associate with youth, having an excess of systemic tyrosine or hyperactivating its signaling pathways relentlessly drives the biological gears of aging. Let us look at why less tyrosine might actually mean more time. Conventional nutritional wisdom dictates that amino acids are universally beneficial, required to maintain muscle mass and initiate cellular repair. Yet, longevity research consistently proves that protein restriction, and specifically the restriction of certain amino acids, can significantly extend lifespan across multiple species. Recently, the scientific spotlight has turned intensely toward tyrosine. A groundbreaking cohort and Mendelian randomization study, leveraging massive datasets from the UK Biobank, fundamentally shifted our understanding of this amino acid. The researchers investigated the impact of circulating levels of phenylalanine and its downstream product, tyrosine, on human lifespan. The results were striking. Genetically mimicked higher circulating tyrosine levels were independently associated with a shorter lifespan. Intriguingly, this effect exhibited a distinct sex disparity, showing a highly significant correlation with reduced longevity in men, even after controlling for phenylalanine. But why would a naturally occurring, essential adjacent amino acid shorten your lifespan? The answer lies in the ancient, evolutionary nutrient-sensing pathways that govern all cellular metabolism. Your circulating tyrosine levels are intricately tied to the insulin and insulin-like growth factor 1 signaling pathway, as well as the mechanistic target of rapamycin complex 1, commonly known as mTORC1. When tyrosine is abundant in your bloodstream, these pathways are continuously stimulated. Think of the mTORC1 pathway as a biological cellular foreman. When it senses nutrient abundance, it orders the cell to prioritize growth and reproduction over repair and maintenance. Over decades, this relentless push for cellular growth exhausts your vital stem cell pools, accelerates cellular senescence, and drives the physical phenotypes of aging. By genetically or dietarily restricting tyrosine, these nutrient-sensing pathways are powerfully suppressed. This suppression mimics the exact molecular effects of caloric restriction, shifting your cellular machinery into a highly conserved survival and repair mode that effectively slows down the entire aging process. But if high circulating tyrosine accelerates aging, how exactly does the body manage its tyrosine economy? And more importantly, can this process be hacked for our benefit? Pioneering research utilizing Drosophila melanogaster, or fruit flies, has provided a fascinating glimpse into the metabolic reprogramming associated with extreme longevity. In aging organisms, there is a natural, age-dependent increase in the specific enzymes responsible for the tyrosine degradation pathway. Under normal circumstances, your body breaks down tyrosine to feed the mitochondrial tricarboxylic acid cycle, also known as the TCA cycle, for basic energy production. However, researchers discovered that in naturally long-lived genetic variants of flies, the metabolome is drastically altered, featuring much higher intracellular pools of preserved tyrosine. To test this mechanism directly, scientists genetically downregulated tyrosine aminotransferase, or TAT, which is the first and rate-limiting enzyme in the tyrosine degradation pathway. The results were astonishing. Both whole-body and neuron-specific suppression of TAT significantly extended the fly's lifespan. By preventing the breakdown of tyrosine for basic mitochondrial fuel, the cells were forced into a metabolic detour. This specific cellular reprogramming redirected the preserved tyrosine toward the synthesis of vital neuromediators like dopamine and octopamine, which typically and detrimentally decline with age. Furthermore, blocking this degradation pathway mitigated mitochondrial dysfunction, specifically by rescuing the age-related suppression of the electron transport chain complex 1. This presents a beautiful biochemical irony. While high systemic circulation of tyrosine from the diet stimulates pro-aging mTOR pathways, restricting its intracellular degradation preserves the amino acid for neurological maintenance and forces a beneficial metabolic stress response that drastically extends lifespan. To truly understand tyrosine's powerful grip on the aging process, we must look at how it functions within the actual architecture of proteins. Tyrosine possesses a phenolic hydroxyl group on its molecular ring, making it a prime target for kinases. Kinases are highly specialized enzymes that attach a phosphate group to this ring. Receptor tyrosine kinases, or RTKs, are transmembrane proteins that act as vital antennas for the cell. When a growth factor, like insulin or IGF-1, binds to the outside of the receptor, the RTK activates and phosphorylates its own tyrosine residues on the inside of the cell. This newly phosphorylated tyrosine acts as a physical docking station for other proteins, initiating a massive cascade of signals that reach deep into the nucleus of the cell. The insulin and IGF-1 signaling network, heavily reliant on these precise tyrosine phosphorylation events, is the most universally conserved aging pathway in all of biology. Attenuating this pathway extends lifespan in yeast, worms, flies, mice, and almost certainly humans. However, the RTK family is vast, and researchers have begun hunting for other specific tyrosine kinases that might be driving the aging process in the background. Enter anaplastic lymphoma kinase, or ALK. Traditionally studied almost exclusively in the context of nervous system development and oncology, the role of ALK in aging was, until very recently, a complete mystery. Recent breakthrough studies have revealed that inhibiting ALK signaling, whether through targeted genetic mutation, RNA interference, or by targeting it with small molecule inhibitors like TAE684, extends healthy lifespan and preserves critical neuromuscular function in aging organisms. Furthermore, reducing ALK signaling specifically in adult neurons promotes extreme resistance to starvation and xenobiotic stress, and remarkably improves sleep consolidation, a physiological function that famously deteriorates as we get older. The localized inhibition of neuronal ALK appears to modulate the organism-wide expression of insulin-like peptides. This proves that dialing down specific tyrosine kinase signaling locally in the brain can orchestrate a massive systemic anti-aging effect across the entire body. The pharmacological implications of this are immense. Kinase inhibitors, which are currently a cornerstone of targeted cancer therapies, are now being viewed through the entirely new lens of geroscience. By precisely fine-tuning the activity of specific tyrosine kinases, we may one day be able to pharmacologically mimic the profound longevity benefits of genetic mutations. If kinases are the molecular switches that turn aging signals on, protein tyrosine phosphatases, or PTPs, are the biochemical erasers that remove the phosphate groups, turning the signals off. The incredibly delicate equilibrium between kinases and phosphatases determines the youthful homeostasis of a cell. As we age, this critical balance is profoundly disrupted, leading to a biological phenomenon known as cellular senescence. Senescent cells are essentially zombie cells that have ceased dividing but aggressively resist programmed cell death, known as apoptosis. They accumulate in aging tissues and secrete a highly toxic cocktail of pro-inflammatory cytokines known as the senescence-associated secretory phenotype, or SASP. This SASP damages surrounding healthy tissue and rapidly accelerates the aging of the entire organ. Recent investigations have highlighted the critical role of protein tyrosine phosphatase 1b, or PTP1b, in this exact degenerative process. In human chondrocytes, which are the specialized cells responsible for maintaining joint cartilage, the painful progression of osteoarthritis is heavily driven by cellular senescence. Researchers have discovered that PTP1B acts as a major facilitator of this senescence. PTP1B normally functions to dephosphorylate and inactivate the insulin receptor, and its overactivity is a known primary driver of metabolic dysfunction and severe insulin resistance. Strikingly, genetically inhibiting or knocking down PTP1B dramatically alleviates chondrocyte senescence, drastically reduces the inflammatory SASP, and enhances longevity-associated pathways. This powerful anti-aging effect is intricately linked to SIRT1, the famous longevity protein widely known to be activated by molecules like resveratrol. SIRT1 naturally downregulates PTP1B. Thus, selectively activating SIRT1 or pharmacologically inhibiting PTP1B represents a highly potent therapeutic strategy for reversing age-related tissue degeneration and completely restoring cellular insulin sensitivity. Another fascinating player in this arena is SHP2, a ubiquitously expressed tyrosine phosphatase that operates not just in the fluid cytoplasm, but crucially within the nucleus of the cell. Telomere shortening is a primary hallmark of human aging, normally counteracted by the vital enzyme telomerase reverse transcriptase, or TERT. Under conditions of age-related oxidative stress, rogue SRC kinases inappropriately phosphorylate TERT on its tyrosine residues. This forces the physical ejection of this protective enzyme from the nucleus, leaving the cellular DNA highly vulnerable to rapid aging and degradation. Nuclear SHP2 steps in as a dedicated molecular defender, rapidly dephosphorylating TERT and completely preventing its nuclear export. In this microscopic battleground, the preservation of cellular youth depends entirely on the cell's ability to swiftly remove rogue tyrosine phosphorylations before they can trigger the irreversible mechanisms of aging. Perhaps the darkest side of tyrosine's biochemistry lies in its extreme physical vulnerability. The exact same phenolic ring that makes tyrosine a perfect biological signaling switch also makes it highly susceptible to catastrophic chemical attack from reactive oxygen species and reactive nitrogen species. As an organism ages, accumulating mitochondrial dysfunction and chronic low-grade inflammation lead to a devastating storm of free radicals. Tyrosine acts as a physical cellular sponge for this intense oxidative stress, becoming irreversibly scarred in the process. When exposed to peroxynitrite, a highly destructive free radical formed by the volatile reaction of nitric oxide and superoxide, the tyrosine residues on your proteins are permanently converted into 3-nitrotyrosine. Similarly, hydroxyl radicals can alter the standard paratyrosine into highly abnormal isomers like metatyrosine and orthotyrosine, or even chemically cross-link two independent tyrosine molecules together to form a dysfunctional structure called dityrosine. These chemically modified tyrosines are not merely harmless biological markers of getting older. They are actively pathogenic. When a vital protein's tyrosine residue is nitrated or oxidized, its complex three-dimensional structure is violently warped, and it can absolutely no longer be properly phosphorylated by kinases. A prime example of this failure is the 3rd of March 14 protein family, which regulates a vast and critical interactome of cellular survival processes. The oxidative conversion of specific tyrosine residues, like Y130 and Y213, to 3-nitrotyrosine on the 3rd of March 14 protein completely destroys its structural ability to bind to client proteins. This results in a catastrophic dysregulation of essential downstream survival signals. Furthermore, dietary exposure to and the systemic accumulation of these oxidized tyrosine products directly promote massive tissue fibrosis. Prolonged cellular exposure to oxidized tyrosine aggressively activates the MAPK and TGF-beta1 signaling pathways in the liver, effectively driving the irreversible conversion of healthy metabolic tissue into rigid, non-functional fibrotic scar tissue. Similarly, the bodily accumulation of metatyrosine and orthotyrosine has been heavily and directly linked to systemic insulin resistance, rapid cardiovascular decay, and the onset of both acute and chronic inflammatory states. In essence, the oxidative modification of tyrosine is the exact biochemical equivalent of literal rust accumulating on the microscopic gears of a biological clock, causing the machinery of life to violently grind to a halt. To view the devastating impact of tyrosine biochemistry on a truly macroscopic scale, one must simply look to adipose, or fat, tissue. While often incorrectly dismissed by the public as mere inert energy storage, adipose tissue is actually a highly active endocrine organ and is increasingly recognized by science as a primary pacemaker of organismal aging. As we age, our adipose tissue undergoes profound negative changes, heavily accumulating those senescent zombie cells that secrete highly inflammatory SASP factors into the blood. At the deepest molecular level, one of the earliest and most severe functional dysfunctions in aging fat is the catastrophic failure of the insulin signaling cascade. This failure is specifically characterized by a severe and measurable reduction in insulin-stimulated tyrosine phosphorylation. Because the insulin receptors in aged adipocytes can no longer efficiently undergo necessary tyrosine autophosphorylation, the cells essentially become deaf to insulin in the blood. This localized insulin resistance isolated in fat tissue quickly creates a massive systemic ripple effect, aggressively promoting total body lipotoxicity, chronic low-grade systemic inflammation, severely impaired glucose tolerance, and widespread metabolic syndrome. Thus, the simple loss of proper tyrosine kinase signaling in just one single tissue type can forcibly drag the entire human organism into an accelerated state of aging. The story of tyrosine biochemistry perfectly encapsulates the intricate, multi-layered, and fascinating nature of cellular longevity. It is a molecule defined by its incredible dualities. It is absolutely essential for synthesizing the dopamine that keeps our brains sharp, motivated, and youthful. Yet it is fully capable of driving the mTOR pathways that accelerate systemic bodily decline. It acts as the absolute critical on switch for insulin and growth factor signaling, yet its sheer physical vulnerability to oxidative modification allows it to become a primary agent of cellular fibrosis and irreversible senescence. The entire future of gerotherapeutics will heavily rely on manipulating this incredibly delicate balance. We are rapidly approaching a new era of biological understanding, where precision medicine will intercept aging at the exact level of the tyrosine residue. Interventions of the near future may include dietary and metabolic modulation, utilizing advanced precision nutrition to closely manage systemic tyrosine levels, thereby keeping the pro-aging mTORC1 pathway in check while ensuring enough vital precursors remain available for total brain health. We will see the rise of next-generation kinase inhibitors, brilliantly repurposing highly specific tyrosine kinase inhibitors, such as those targeting the ALK receptor, to successfully suppress pro-aging signaling pathways in a highly tissue-specific manner, all without causing the severe, toxic side effects typically associated with blunt force oncology drugs. We will also see the development of novel phosphatase activators, utilizing highly specialized compounds that selectively modulate PTPs. These will inhibit the senescence-driving PTP1B while simultaneously enhancing the critical telomere-protecting actions of nuclear SHP2. Finally, we will see the implementation of anti-nitration scavengers, utilizing hyper-targeted antioxidants and advanced anti-inflammatory agents to specifically and physically protect the vulnerable phenolic ring of tyrosine from being converted into toxic 3-nitrotyrosine or dityrosine, successfully preserving the physical structural integrity of the entire cellular interactome. The deep biochemistry of tyrosine proves once and for all that the aging process is not an inevitable, chaotic breakdown of biology, but rather a highly regulated, elegantly orchestrated signaling program. By precisely deciphering the complex language of tyrosine phosphorylation, oxidation, and degradation, we are learning not just how the biological clock ticks, but exactly how to reach inside the machinery of life and gently rewind the hands. Thank you for joining us for this deep dive on Fun Facts Online.